Hi everyone, we're from Group 1 and today we're going to present on Botulism or Zombie Bombie for our PBL assignment, and we are proudly supervised under Prof. Dr. Siti Suri Arshad.
For case review, it started with Bella and Anna, which are primary school siblings, having similar clinical signs. This includes difficulty swallowing and breathing, as well as blurred vision for 2 days. They met traditional healer, but had worse clinical signs after the treatment. Then they were brought to the emergency department. Supportive treatment and diagnostic investigation were done. Unfortunately, Anna died within 4 hours due to respiratory failure. Anna's postmortem turns out no abnormal findings. However, the blood result shows positive for botulism toxin, which matched with the antitoxin test done in backyard chicken, which also positive for botulism toxin.
So, what is botulism? So, botulism, caused by the bacteria Clostridium botulinum, it is a life-threatening disorder from the poisoning where it releases its neurotoxin and affecting our peripheral nerves. The main clinical signs are seen as weakness or paralysis of the limbs, difficulty in chewing or swallowing, shortness of breath, and death is usually caused by respiratory arrest in this case. So this falls under the BSL Class 2, where there's actually restricted laboratory access to this organism, where we will need appropriate PPE to protect our eyes and face, and must be conducted in a biological safety cabinet, and waste must be decontaminated before disposal.
So, how humans get botulism? Firstly, it is common from a foodborne transmission such as consume infected poultry or contaminated fish that contain botulinum neurotoxins. Consumption of maggot from decaying carcasses of infected birds can also pose a risk, and also due to improper cook of chicken meat. In a rare case scenario, human also could be infected through waterborne transmission. For the epidemiology part, we will start with agent that cause this disease, which is Clostridium botulinum. There are 3 major strains of Clostridium botulinum produce neurotoxins. For the type C and D botulinum toxin, it will affect avian and non-human mammalian botulism, while type A, B, E, and F will cause human food poisoning disease. The hosts that are highly susceptible for this agent are poultry, particularly chickens, turkeys, ducks, and waterfowl. Factors that affecting the host to get this disease are ingestion of contaminated materials, contaminated feed, litter, boots, and equipment have also been reported as possible sources for introducing Clostridium neurotoxin into a poultry farm. Carcass maggot cycle, where the maggots get the botulism toxin through the ingestion of the carcasses, and the maggot can considered as a mechanical vectors where they can store various botulinum neurotoxins. There are several types of environment that enhance the growing of this agent, which are anaerobic conditions such as stagnant water, decaying vegetation, and carcasses provided low oxygen environments. Next is a high organic matter where decomposing place of decomposing fish, birds, or other carcasses promote bacterial growth. And lastly is the warm temperatures environment. Botulism is more common in warm, humid conditions that accelerate decay.
Moving on to the diagnostic methods. So first is the clinical history. Clinical history can be obtained from the patient to acquire possible risk factors. Second is the ancillary test, which includes electromyography, hydrophonium chloride test, brain imaging, and also cerebrospinal fluid examination. Thirdly is the laboratory test, which is often confirmatory for botulism diagnosis, which includes mouth bioassay, ELISA, and real-time PCR, which is used for toxin demonstration from stool, serum, or food. Bacterial culture can also be done. However, it is important to note that it's very difficult to culture Clostridium species. So what is electromyography? So electromyography helps to measure the muscle response or electrical activity due to nerve stimulations to the muscle. So this test is basically used to help detect neuromuscular abnormalities, which is also seen in this case.
Hydrophonium chloride test, or known as Tansilin test, is used to diagnose myasthenia gravis. It usually negative in botulism patient, however, false positive might be occur in some botulism cases. Mouse bioassay is the gold standard method for identifying botulism neurotoxin, and this is the only FDA-approved method for laboratory confirmation of botulism. Specimens are injected intraperitoneally into the mice with and without antitoxin. However, low levels of toxin that are sufficient to produce human illness might not produce signs in mice.
So for laboratory tests for botulism, there are two types of tests. The first one, which is ELISA test, that used to detect botulism neurotoxins. And for the second one, which is real-time polymerase chain reaction, which is RT-PCR, it is applied in the qualitative detection of Clostridium botulinum type A, B, and E. So for real-time PCR, it can be used to detect both bacteria and also neurotoxin itself. On a side note, in this case, how alkaline water contribute to Clostridium growth? Clostridium cannot grow in acidic condition, and one study shown that there's a peak growth at pH 9, which is alkali, and it start to grow at pH 7, which is neutral. So this is why I think it can be related in our case where the alkaline water contributes to Clostridium growth and leads to Clostridium infection in the backyard chicken farm.
So the farm management is the first line of defense against botulism. So under the environmental management, animals must be provided with fresh, uncontaminated food because wet or spoiled feed can create the anaerobic conditions ideal for bacterial growth. And then the stagnant water, another potential source of spores, must be replaced with clean, fresh water daily. The next one is disease treatment and biosecurity measures. So in terms of biosecurity, it begins with the first one, the regular cleaning and disinfection of feeding equipments, water containers, and animal droppings to minimize the contamination itself. Next one is vaccination programs. So vaccinations really help reducing their susceptibility to botulinum toxin, providing an added layer of protection against outbreaks, and it must be implemented by farm workers, or maybe the farm workers itself can train their staff on feed management, disease recognition, and biosecurity practices.
Next, in the case when we want to dispose of chicken carcasses that have botulism in Malaysia, here are the recommended practice regarding distance and depth for the burial. Firstly, the carcasses should be buried at depth of at least 1.5 meters. This depth helps prevent scavengers from accessing the carcasses and minimize the risk of contamination of surface water. Secondly, the burial site should be located at least 30 meters away from any farm buildings, water sources, or area where animal graze. This distance helps to reduce risk of the disease transmission and also contamination. So, these are 5 keys to safer food from WHO, which were developed to educate safe food handling behavior to all consumers and food handlers. First, keep clean by wash your hands before handling food and often during food preparation. Secondly, separate raw and cooked raw food, especially meat, poultry and seafood and their juices. Thirdly, cook thoroughly your food. Fourth, keep food at safe temperature. Lastly, use safe water and also raw materials, and please select fresh and wholesome food.
Contact tracing is a process of quickly identifying, assessing, and managing people who have been exposed to a disease to prevent additional transmission. This includes 3 key steps. Initially, we should conduct a disease transmission experiment. This experiment will analyze disease test results and draw a map to identify the index cases for the disease outbreaks. By doing so, you should be able to trace this outbreak back to its source. Next, plan a contact tracing interview. Then finally, share your findings. One of the ways CDC communicates information is through social media. And current advancements such as digital contact tracing and genetic sequence data in contact tracing successfully helps to reduce the transmission rate of disease.
Since botulism is a notifiable disease according to DVS, we need to notify DVS regarding cases of botulism. Ministry of Health Malaysia MOH is responsible for monitoring and responding to foodborne illnesses, including botulism. Food Safety and Quality Division, which is under MOH, oversees food safety regulations and conducts inspections. DVS plays a critical role in monitoring animal health and ensuring food safety standards in poultry farming. WHO provides international guidelines and support for managing foodborne diseases. To prevent spread of the disease, we should do awareness programs such as short videos, community networks with NGOs, as well as media campaigns. One Health collaboration is important in emphasizing the interconnectedness of human, animal, and environmental health, which is crucial for addressing botulism.